Okay, hi there. Welcome everybody to the Ataxia Global Young Investigator Initiative webinar series. As you know, the Ataxia Global Initiative is hosting this webinar series for young investigators in the ataxia field to introduce you to important aspects of various research topics. In the first webinar series of the Ataxia Global Young Investigator Initiative, each AGI working group will present an introduction into their topic. Thomas Klockgether studied medicine at the University of Göttingen, and during this time he also carried out research at the Max Planck Institute for Experimental Medicine. After graduating, he went to Oldenburg for clinical training and then returned to the Max Planck Institute to work in basic research on Parkinson's disease. He completed his neurology training in Tübingen, where he also began to focus on degenerative ataxias, in addition to pursuing research on Parkinson's disease. These research lines evolved very successfully during his appointment in Bonn as a chairman of the Department of Neurology, and Professor Klockgether has been the Dean of the Medical Faculty of the University of Bonn from 2008 to 2011. And from 2011 to 19, he has been speaker for the Center of Rare Diseases in Bonn, and since May 2011, he is director of the clinical research at the DZNE. Professor Thomas Klockgether initiated pioneering cohort studies in ataxias, such as the EUROSCAR-RISCA cohort and the ESME cohort. He gave his initial impulse to form a global initiative and is now chair of the Ataxia Global Initiative. Scope of this webinar is to introduce and present a standard clinical assessment program for ataxia studies. A focus will be on the discussion of the strengths and weaknesses of available clinical scales, and the seminar will provide an outlook on further development of scales and their use in upcoming clinical trials. With this, I would like to thank you for being here today and enjoy the talk.
Yeah, hello everybody, and thank you for the kind introduction. So the talk is on clinical outcome assessments, and my first slide shows the learning objectives, and participants by the end of this webinar should be able to apply ataxia staging systems. They should know the most important properties of two clinical ataxia scales, the SARA and the FARS, should be able to discuss the relevance of INAS, understand the use of performance-based tests in ataxia, and assess the importance of patient-related outcome measures. And according to these learning objectives, the outline the agenda of my talk. We start with staging systems, then discuss SARA and FARS, then introduce the quantitative performance measures for ataxia, and finally deal with the patient-related outcome measures. After each of these parts, there will be one multiple choice question. I will give recommendations for use of these outcomes, scales, questionnaires, and so on. And these recommendations are not my personal recommendations, but they are rather based on the consensus of the Clinical Outcome Assessment Working Group of the Ataxia Global Initiative. So I will start with staging systems. Why do we need staging systems? They give a very broad, not very detailed information, so they are not useful as outcomes for clinical trials because they lack precision and details, but they give a very relevant information about the status of the patient, and they may be useful for the recruitment of patients. Maybe you plan a trial where you want to have patients in an early disease stage, and you can simply define that by stage 1 of this or that ataxia staging system. This is one staging system, maybe the first one, I believe, that we developed in Tübingen in the 90s. And this is mainly based on Walking ability has 5 stages, ranging from stage 0, which is healthy state without gait difficulties, then going to stage 3, confinement, to wheelchair, and also includes the final stage, which is death, so a 5-stage system. And this is a very simple staging system, as you can see. The advantage of this, it's very robust, so it's very clear whether you are in stage 3 or stage 4. There is no doubt about the distinction, and this can be applied by interviews, even by telephone interview. And the original publication, you see 2 figures of this original publication that appeared in 2098, was completely based on telephone interviews that were done by students. That was at that time a very large group of almost 500 ataxia patients. And with this simple instruments, we could, maybe we were the first to show that SCAR1 has a faster progression than SCAR2 and 3, something that was then later confirmed in many prospective longitudinal studies. Another stagings we have recently, and this is a work of Heike Jacobi, that just yesterday appeared in the Annals of Clinical Translation Urology, where you can download the paper. We have reused this staging system and applied it to the RISCA and EuroSCAR data. Again, a very large cohort together of 600-700 participants, and you can see that there is increasing ataxia severity, not a very unexpected, here measured with a SARA, but the different stages from 0 to 3 are clearly defined, and there's little overlap of the SARA scores. And we did a multi-state modeling, quite complicated biostatistics, but this allows to define the probability of a certain person with a SCAR1, 2, 3, or 6 at a given age to be in a certain age. For example, if you go to SCAR1 at the age of 40 years, you can say that a SCAR1 patient has a probability of 25% to be in stage 1, of 30% to be in stage 2, and of 10% to be wheelchair bound. This multi-stage modeling also allowed to define and estimate the length of duration. So on the basis of this analysis, we can say the average time a SCAR1 patient stays in stage 1 or stage 2 is 8, 9, 10 years. You can read this, all the details in this publication. In 2006, the paper in Urology, first author Lynch, appeared from the US Ataxia group, and this was a paper on the Friedreich's Ataxia rating scale, and the Friedreich's Ataxia rating scale has 4 parts, and part 1 is another functional staging system. And you can see here that this is more detailed, more fine-graded. So there are 7 stages from 0 normal to 6 confined to wheelchair. And there is the possibility of increments of 0.1. So if you feel that a patient is just in between stage 3 or stage 4, you can rate it with 3.5. So the advantage of the FAST part 1 compared to the older staging system is the more higher precision and more details. On the other hand, if you're looking to stage 1, minimal signs detected by physician during screening. So this requires a examination. This cannot be applied by a simple questionnaire or telephone interview. So this was discussed in the working group, and the majority of the working group preferred the fast functional staging that I just showed you. So the recommendation is to use the fast functional staging for ataxia at every routine visit. So not only in observational or interventional trials, but to document the fast functional staging for ataxia at every routine visit in the clinic or the hospital. So I continue with the first multiple choice session. Which of the following statements is correct? There's only one answer is correct. The staging system introduced by Klockgether et al. Is finer graded than the fast functional staging. Second statement, the fast functional staging can be easily applied by telephone interview. Staging systems are useful as outcome measures in clinical trials. The staging systems used in ataxia are mainly based on walking disability. And fifth statement, the correct use of ataxia staging systems requires long-standing clinical experience. And now you have 1 minute or less time to give your answer. And Anna-Marie will then post the results.
Yes, so quite some people are already answering this question. We'll leave it open for a bit more. And I think this is a representative view of the public's opinion.
The correct answer is 4. The staging system used in ataxia are mainly based, they're not fully based because death is not based on walking disability, but they are mainly based on walking disability, and the majority gave the correct answer. Answer number 3, staging systems are useful as outcome measures in clinical trials. I think this is not completely wrong because you can design very simple trials where you treat patients over a longer time period and then simply look whether they become wheelchair bound or not. A very robust and simple design that is principally possible. It's not very realistic because such a design would need many years for trials, so it will not be applied by pharmaceutical industry. But again, answer 3 is not completely wrong. Okay, I think we can continue, and I will continue with the clinical ataxia scales, which may be the most important instruments as outcome measures. And this is an incomplete list of ataxia scales, but nevertheless, the scales you can see here are the most important and those which are most frequently used and are still in use. The first scale that was published is the ICARS. You can see here the number of items, and compared with the SARA, it has more than double number of items. This immediately shows that it's more time consuming, more complicated. Then in 2006, we developed and validated the SARA, which is now very commonly used. And it was initially developed for spinocerebellar ataxia, but then we did a number of additional validation trials in Friedreich's ataxia and in other types of ataxia, so that here we really have evidence that this is a scale for ataxia in general and not related to a specific ataxia disease. This is a difference to the FARS that I already mentioned. The FARS was published in the same year, even the same issue of Neurology in 2006, with 4 parts. I already introduced the first part, the staging system, and here the third part is the conventional clinical scale. So again, you can see that it's much longer, it's more time-consuming than SARA, and it's specific for Friedreich's ataxia. So it does not make much sense to use the FARS clinical scale for spinocerebellar ataxia patients or ataxia-telangiectasia. The same applies to the NESCA, which is used mainly in Brazil and other Latin America countries. This is specifically related to SCA3. And there's another quite useful, very short scale, the brief ataxia rating scale, only 5 items, which was developed and published by Jeremy Schmarman. I will focus on the 2 deemed most important, one, the SARA and the clinical part of the FARS. The SARA, I assume that most of you know SARA quite well and maybe even applying it quite frequently. Nevertheless, I would like to give a general description of this scale. It's, as mentioned, the clinical rating scale based on a standard neurological examination. So you need some neurological expertise and skills, but no specific skills. And the 8 items are related to gait, stance, sitting. So the postural axial movements, then the speech, and then 4 additional items related to arm and leg movements. And I already mentioned that we did a number of validation trials, not only in the spinocerebellar ataxia, but also in Friedreich's and other ataxias. And here on the right, you can see some results of the initial validation study showing a high degree of reliability, evidence for validity, and we also provided data suggesting linearity and sensitivity to change of the scale. 2 additional information. The SARA is completely free. You can download it from this website. I hope that in the meantime, it's also available on the AGI website. So this is the old Ataxia Study Group website. So you can download it, freely use it. I still have 1 mail per week where physiotherapists or rehabilitation clinics ask whether they can use it, or the pharma companies, whether you can use it for trial. There's no licensing fees. You can download it and use it. And if you want to use it properly, I strongly recommend to do a training where you can also receive a certificate of a trained SARA rater, and this is available and free for Ataxia Global Initiative members, and you find the links to it on the AGI website. The next slides, I will briefly go through the examination procedure. So it starts with gait. Ideally, you should have a 10-meter hallway where the patients can walk. Should be done in about 50 centimeters distance to the wall, and as always, safety first. So the examiner should be close to the patient to prevent falling or any injuries. This is then the tandem gait, which to be rated as positive needs to be done without any help. So this is only for demonstration. This woman does not perform the gait, the tandem gait properly. The next is the stance. Here, patients have 3 trials, and the best of the 3 trials is rated. And the task is to stay in the defined position with feet apart or with feet together or in tandem stance for at least 10 seconds. Then the sitting. For a sitting, you should have an examination bed where the patients should not touch the ground with their feet, and then the degree of stability is rated. Now I'm continuing with the items related to arm movements. This is the finger chase, and Jennifer does it in the correct way, very fast movements in unpredicted direction. And what is rated is the dysmetria. And this is in many cases, as in the present case, it's an overshoot, but it may be also undershooting. And what you're rating is the distance of the patient finger after the first movement to the intended goal. And then the nose finger test. And here you can observe all aspects of ataxia, but what is rated here is only the tremor. So you do not consider how smooth the movement is. It's only rated the degree of this oscillatory movements of the tremor. Then the fast alternating movements, again a timed test. It's rated how precise the movements are made and whether the participants perform within the maximal time of 10 seconds. And finally, the heel-shin slide. Here, patients have no view on their legs. They should do it without trousers, so as demonstrated here, and what is rated, how often the heel leaves the shin during the 3 trials. So in the next slide, I'm showing one of the applications. So Sarah is now almost routinely used in observational longitudinal studies. It's also used in interventional trials, and this is from the EuroSCAR study, a longitudinal study of SCAR 1, 2, 3, and 6. And these participants were followed over 8 years. And you can see what I already suggested on the basis of the data of the validation trial, that there is linearity of the scale. So it's a linear increase over time for SCAR 1, 2, 3, and 6. You can also see that SCAR 1 has the fastest progression. SCAR 2 and 3 are almost identical and intermediate, and SCAR 6 has a slower progression. There is an ongoing debate on SARA. SARA is being used in clinical trials. It is commonly accepted by the European Medical Agency, the EMA, but it was criticized by the US agency, the FDA, and they believe what I do not, where I do not agree with. They believe that all the arm items, so the finger pointing, the fast alternating movement, are irrelevant for patients. This resulted in modified but unvalidated versions of SARA, which are currently used at least in one clinical trial, the Biohaven trial in the US, where a shortened, modified version of SARA is used that includes only the first 4 items, gait, stance, sitting, and speech. Now the Friedreich's Ataxia rating scale. I mentioned that this is a quite long, complicated scale with 23 items. This is the original scale, and FARS already underwent a modification, and this modification was a shortening. And here on the right side, you can see this shortened, modified version of FARS that was published in 2019. And what you can see here is it was made very similar to SARA, because all the Friedreich ataxia specific items related to peripheral nervous system involvement were taken out, and same with the bulbar involvement, and what remains is ataxia, and the ataxia tests nose finger dysmetia, rapid movements, and sitting, stance, walking are very similar to SARA. So now with this new modified version, the modified version is very simpler, still longer, but very similar to the SARA. The modified version of Friedreich's of the FARS was used in a positive clinical trial, the MOXIE trial, testing a compound named omaveloxolone that was published and is positive, first positive trial for Friedreich's ataxia. Again, the outcome measure was the change of the modified FARS from baseline, and you see that in the treatment group there was an improvement over 1 year, 48 weeks of 2 points compared to the placebo group that worsened by 1 point. This is currently in the process with the FDA, so the FDA is not fully accepting the data. They want to have additional data, but as I understand, there's still hope that omaveloxolone will be approved in the US and maybe later then also in Europe and other countries. What I've not yet mentioned is the INAS, the Inventory of Non-Ataxia Signs. This is not a scale. This is a list of non-ataxia symptoms, and I discussed the scales and said that there are some scales who are focusing on the ataxia like SARA and also the modified version of the FARS, and others have additional symptoms, in particular the NESCA. There are all kinds of items related to dystonia, Parkinsonism, all these things that can happen in SCA3. So when we designed SARA, we took the very clear decision that we wanted to have a pure ataxia scale. On the other hand, we saw the need for having some information about the non-ataxia symptoms that can appear in spinocerebellar ataxia, but also other ataxia disorders. And this is the background of INAS. So it's a simple list that can be obtained by a routine clinical examination, urological examination, and you can quantify that and then produce the INAS count, which ranges from 0 to 16. So 0 high means that there are no non-ataxia symptoms, and 16 is the theoretical maximal involvement of other symptoms outside those which are associated with ataxia symptoms. So we also have used the INAS in the Euroscar study, again, the data from the 8-year follow-up. And you can see nicely here that there is this increase. Again, it is a bit faster in SCAR1 compared to SCAR2 and 3, but this is not a linear increase. We observed for the INAS some plateauing, so after 4 or 5 years there was no further increase, and showing the strength of INAS, you know that SCAR6 is the prototype of a spinocerebellar ataxia that is, as we say, purely cerebellar or purely ataxia, and indeed the INAS scores of SCAR6 are much lower from the beginning than those of SCAR1, 2, and 3, and there's almost no increase. And that's what you can see here as 1 or 2 INAS points in SCAR6 is mainly due to the higher age of the patients and not part of the clinical spectrum of SCAR3. Again, the recommendations from the working group. So for the routine clinical examination, not any observational study, just your routine visit at the ataxia clinic, perform a complete neurological examination. You do not need to do the INAS, just do a neurological examination, but use the SARA at every routine visit. For observational studies, there's the clear recommendation to use the SARAs and INAS, and SARA was the preferred scale compared to the FARS because this can be applied to all ataxia types, and in your outpatient clinic you typically have free drug patients, SCAR patients, unexplained ataxia patient, and so on. And it makes sense to have one scale for all rather than switching between FARS, SARA, and NESCA, dependent on the specific diagnosis. So again, I will continue with multiple choice. Which of the following statements is wrong? Now the question, which is wrong? And again, one of the statements is wrong. I read them. SARA has 8 items. FARS has been specifically developed for use in free drug ataxia. SARA has been validated in various ataxia disorder. INAS is useful to assess non-ataxia symptoms in spinocerebellar ataxias. And 5th statement, INAS requires specific instrumentation. Now it's open for your answers.
In the meantime, maybe there's a short question about INAS. The question is anxiety a part of INAS?
No. Here in INAS, we are focusing on neurological symptoms. In my last part on the patient-related outcomes, I will mention the mental symptoms, including anxiety.
Thank you. Then we can also end the poll.
Yeah, the vast majority of you is completely right. INAS does not require specific instrumentation. It's just on the basis of a simple neurological examination. You need a reflex hammer and a vibration fork, but this is no specific instrumentation. Okay, we can continue then with the quantitative performance measures, and here I'm talking about timed tests, which indeed require specific instrumentation. The first who presented the use of such timed performance measures was the Friedreich group. It's the fourth part of the FARS with a 9-hole pegboard test and the PATA rate. We developed a very similar instrument that also includes the 8-meter walk. We named it SCAFI, and it combines these 3 tests, and there's a third quantitative performance measure, the CCFS, that was developed in Paris. This is from the original publication introducing the SCAFI, the SCA functional index. As I mentioned, it is based on the PATA rate, just counting the number of PATA syllables within 10 seconds, then the well-known 9-hole pegboard test and an 8-meter timed walk. For all these time tests, it's very important to be very closely and precise, follow the instructions, because there are various options to perform the 9-hole pegboard test, and unfortunately in different studies and trials there are different instructions. So if you are participating in a study, whether observational or interventional, and there are these tests are used, including the 9-hole pegboard test, you have to closely follow the instructions, otherwise the results are not useful. For the SCAFI, you have these 3 measurements, and then they are, from these measurements, Z-scores are formed, and they are combined to a single Z-score. This is the SCA functional index. A clear disadvantage of the SCAFI is that you always have to relate it to a normal population, to your control population, and this may vary from study to study. So if you have a SCAFI value of -1.5 or -2, it's always related to the specific control, healthy control population of this study, so you cannot easily compare SCAFI results from one study to another. Now the CCFS, the Composite Cerebellar Function Severity Score, it's different from SCAFI. It's only focusing on the upper extremity. It's a combination on the 9-hole pegboard test and a click test. And these 2 measurements are combined, and you have a score that can be compared between different studies and trials, and you have normal values for each age. So all this measurement is already corrected for age, so this is an advantage. And this now shows how we initially compared the click test with these mechanical devices, and I still find them very useful. In the meantime, some electronic boards were designed, and this is the version that is used in the European Friedreich observational study, the EFACTS, and the most recent version that can be printed by a 3D printer, and that is used in the ReadyScar. And both electronic devices have the 9-hole pegboard and the click test. So when we devised these, the SCAFI and Paris, the CCFS, we were all very enthusiastic, and we believed that we could overcome the subjectivity of clinical scales. But the data we have acquired over time, I must say, are quite disappointing. Here you can see data of a study from Paris, published in 2018, and they simply compare the effect sizes, and the effect sizes tell you something about the sensitivity to change over time. You see here the comparison between ZARA and CCFS. you can see that for SCAR 1, 2, and 3, the performance of SARA is much better than that of CCFS. So for these disorders and diseases, SARA is more sensitive to detect changes. I don't know what happened here with SCAR 7. This is a not very plausible result. We have used the SCAR-V and the CCFS in the RISCA study. The RISCA study was a observational study of non-ataxic risk persons. And here the hypothesis was that we cannot, in these apparently healthy people, that we cannot detect something with our clinical scales, but that these timed measures are already sensitive to pick up changes. But here again, both instruments, SCAR-V and CCFS, lacked sensitivity to change, both in the pre-ataxia RISCA study and the ataxia stages of scars. And this was discussed in the working group, and the recommendation is to consider the use. So if you wish and if you have time, you can use it. But we did not give a recommendation to use these time tests routinely in your outpatient clinics, and even not in observational studies. So it's an option, but it's no recommendation, because unfortunately these measures do not appear to be very useful. Now again, the multiple choice questions related to the timed tests. Now again, I'm asking for the correct answer, which statement is correct. SCAR-V is combined of subtests for walking, speech, fine manual dexterity, and writing. SCAR-V and CCFS are performance-based timed tests. For SCAR-V, an electronic device is available. SCAR-V and CCFS are more sensitive to change than clinical scales, and the CCFS performance is independent of age. And Anna-Marie, you can now again present the questions for your vote.
Quite some different answers are coming in, so I'm very curious to see what the right answer is. Oh, one is coming up as most chosen. So we'll see if that is correct.
Yeah, the second answer, SCAR-V and CCFS are performance-based time tests. This is completely correct, and the majority gave the correct answer. There were 20% voting for statement 1. This was a difficult question, because there's one detail wrong. The SCAR-V does not include writing. It includes the pata, the 9-hole pegboard, and the 8-meter walk. One could include writing, but it's not included. So this was a wrong answer, and the correct answer is statement number 2. And I continue and come to the final part, and that is related to patient-related outcome measures. So these patient-related outcome measures are closer to the patient experience, to their everyday life. I will discuss with a bit of detail the second part of the Friedreich ataxia scale, which is related to activities of daily living, and a quite new questionnaire, the PROM ataxia, that just came out in movement disorders, or will come out within the next weeks. Yeah, start with the FARS part 2, the activities of daily living. This is the original CRF. So it's related to speech, swallowing, cutting food, dressing, personal hygiene, falling, walking, quality of sitting position, bladder function, and it's all related to everyday life. For example, dressing can be normal, somewhat slow, but no help needed. Or then with a rating of 3, with a score of 3, considerable help required, cannot do some things alone. So it's very obvious that these are the things that are very, really important and relevant for the patients. When we started Euroscar, this was not available, and we used the activities of daily living scale from the Huntington disease rating scale, which is similar, which is not identical. And now in most observational studies, this activities of daily living scale from FAS is used. And I think if you have a look at it and see, have a look at the items, one can safely make the statements that this is by no means specific for Friedreich's. This is a general Ataxia, activities of daily living thing. How is it applied? You see here the English version. So it is applied by the doctor, the examiner. So you talk to the patients and try to find the correct answer. The advantage of this is that you can work with the English original version so that you do not need translation into other languages. We found in the Euroscar that was still the Huntington ADL, but nevertheless that there was a linear increase very similar to the SARA of this ADL scale score. And in the Friedreich, the EFACT study, these are, I think, 4-year follow-up data. You can see here on the left the SARA total score, and the entire population is the black line with the black dots. And here on the right side, you see the ADL total score using this scale that I just introduced. And you can see that the annual increase of the SARA is 0.82, and that of the ADL scale is 0.93. And the standard, the variation, the standard deviation is the same. So in other words, the ADL in this 4-year observational longitudinal Friedreich study, the ADL was more sensitive. It's not a big difference, about 10% or so, but it was more sensitive to change than the clinical scale here, the SARA scale. So it appears to be very useful, not only for learning something about the patient perspective, but also for use in clinical trials, because in the clinical trials, you're always looking for the most sensitive instruments. There's a new instrument. The paper has recently come out or will come out, so you can read it in Movement Disorder, the PROM Ataxia. This appears to be very useful, and I'm quite convinced that it will be a standard instrument in the future for many purposes, including clinical trials. But currently, the available data are still very limited, and we do not have any longitudinal data other than for the ADL. And the PROM Ataxia is a questionnaire with 70 items, so it's quite long. And questionnaire means that you hand it over to the patients, and maybe with the support of a help person, but patients themselves fill in the questionnaire. And this means that you need a translation into non-English languages. So this can be done. You can translate it and translate it back. And you should be in contact about that with Jeremy Schmarman so that he can approve the version. You do not need new validation trials, but you need the translation. The Prometaxia, this is the strengths of the Prometaxia, was developed on basis of patient interviews, so it really reflects the problems of the patients. And you have 3 domains. The one is physical domain, and when you are reading the questions, they are very close to that what we are testing in SARA. So are questions related to gait, to speech, to hand movements. Then there's an ADL, which is similar to the FAST ADL, and that was the question that was written in the chat, a number of questions related to mental health, concentration, language, but also including depression and ataxia. So it gives a very comprehensive view of the real world situation patients have with their physical impairments, their impairments with their daily life, but also with their mental problems. So recommendation, these are not instruments that you have the time to use then in your outpatient clinic in routine care, but they should be used in observational studies. The current standard, for example, in ASME and the recessive ataxia registries is the ADL, but in the future, and this is very important to acquire data in the future, maybe it will be replaced by the Prometaxia, because the Prometaxia includes the ADL, but also gives additional information. And with this, I come to the final multiple choice question. And again, you are asked for the wrong statement. So first statement, patient-related outcome measures are become increasingly important in ataxia research. Then in Friedreich's ataxia, the FAST ADL is highly sensitive to change. Then third statement, there are extensive longitudinal PROM data available, PROM ataxia data available. Then among others, the FAST ADL considers speech, swallowing, dressing, personal hygiene, walking, sitting. And final statement, Prometaxia was systematically developed, starting from patient surveys and interviews. And it's open for your vote.
And we're looking for the wrong answer.
For the wrong answer.
Those of you for who it's very late or very early, maybe. All right, the answers have come in.
Yeah, 80% gave the right answer. There are extensive longitudinal PROM data available. They are not available, and is one of the Ataxia Young Investigator group, and maybe, in my view, one of the most important projects of the AGI to acquire these PROM ataxia data. So hopefully in a few years we will know more about that. The first patient-related measures are becoming increasingly important in ataxia research. I think it's more than my personal opinion. If you talk to regulators, to patient lay organizations, they all confirm the importance of patient-related outcomes. And my final slide then summarizes the key points, and I will not repeat and read that again, so that we have still a few minutes for additional questions and discussion. So Anna-Marie, all questions have to be written in the chat, or can you allow?
We can allow people to talk as well. If
Yeah,
someone raises their hand.
Why not?
Maybe before we start the discussion, just a brief note. The patient videos were a little bit scattered, so we all invite you to make use of the Sarah training tool. Annemarie put the link into the chat, so please have a look at that. There's a large library of patient videos of all severity levels, and I think the quality there, of course, will be much better than it can be in such a recorded session. Thank you.
Yeah, the Zoom, I think
Yeah.
the videos on Zoom meetings have poor quality, but these are really high quality videos. And one additional information, all patients specifically gave consent that we can use this video for teaching and training.
I don't see any questions at the moment. Jennifer, Heike, do you have a...
Yeah, I would like to start with a question, since I also did not see any upcoming questions in the Q&A. You started with this disease stages. So on the one hand, we have these disease stages as you introduced in the 90s and the first disease stages. And on the other hand, we have kind of disease phases of pre-ataxic versus ataxic, or pre-symptomatic to symptomatic if we refer to the reported onset of the disease. So what would be your suggestion? How do those stages fit to these disease phases, in particular in the very early stages of normal versus ataxic, or normal versus minimal signs in the first stage?
Yeah, I think in principle, the stages define the disease onset with the onset of a walking disability. And this is the usual definition when you ask patients retrospectively for the ataxia onset. In earlier times we said disease onset, but we know now that the disease already starts many years earlier. So it's the ataxia onset, and that has always in most studies been defined by the onset of walking problems. And so this is just the switch, the change from stage 0 to 1 in both scales. For the early stages, it's very clear that the FARS staging systems gives more information. And if you are particularly interested in the very early stages, the FARS with a more fine-grained information gives more, is more useful.
Thank you. I would have a second question, also regarding your suggestion. What do you think for future clinical trials? Which scales will be more important as secondary outcome parameters? Will that be the ataxia severity, or will that really be the PROM as a patient-related outcome measures? What do you think?
This is very difficult to predict. I think traditionally in most trials in diseases, in particular neurological diseases, whether Alzheimer, Parkinson's, or whatever, Huntington's disease, the primary outcome is a clinical scale. But if you really see that a ADL scale is more sensitive to change, and you know that this is more directly related to patient's life, I would not fully rule out that together with the regulators that make the final decision what they accept as an outcome, it's even, you could even imagine that you have an ADL or the PROM ataxia, whatever, as the primary outcome. But it's very clear that we need more data. The difference between the ZARA and the FARS ADL was very small. In the Euroscar, the ZARA was more sensitive than the ADL scale. And from ataxia, we currently do not have any longitudinal data that allow to discuss this.
There are two questions in the chat. Do you think that a combined ataxia scale gives more information, or should the scales be stay separated?
Yeah, if you mean by combined ataxia scale, whether you combine the FARS with the SARA, this does not make sense. I think you should use one scale and train the scale and do it according to the instructions. If you mean combined ataxia scale, one scale for ataxia in a general, my personal view is that these scales are much more useful than the scales which are specific for one disease, like the Friedreich scale and or the NASCA for SCA3. And I showed with the Friedreich scale that it was modified, and the result of the modification was that they initially had a Friedreich specific scale with all the peripheral things that are typical for Friedreich's ataxia, and now they have a general ataxia scale.
And final question, whether these scales can be applied in patients with motor power less than 4 or 5 on the MRS scale?
Yeah, there is a general principle in the scale, maybe for all scales, but we always definitely say that for the SARA, rate what you see. If someone is completely paralyzed and this person cannot perform the heel shin test, then it's a rating of 4. So worst cannot perform. And you just should rate what you see. It's not interpretation why someone has tremor or why someone cannot sit. So you can apply it to patients in any condition.
All right, I think with that we've come to the end of this webinar. There are no more questions. I would like to thank Thomas Klockgether for his clear overview of these ataxia scales. I think that was very useful.